Okay, good afternoon all. Today I will be discussing the topic that is biophysical chemistry that comes under bioinorganic and biophysical chemistry. So in that biophysical chemistry I will be discussing in the unit 9, that is cell and its constituents. So before getting into the topic, you should be knowing some of the terms. What does the biophysical chemistry says? ಅಂದ್ರೆ ಏನು biophysical chemistry ಅಂದ್ರೆ it is the study of physical properties of biological molecules. So in this paper we are going to study the physical properties, how the reaction will be taking place and what are the effects in the biological molecules, macromolecules will be taking place. So, in the previously, you have studied in the first semester and second semester about thermodynamics in a system, how the rate of reaction will be taking place, and also with respect to chemical kinetics. Whether the reaction is spontaneous or not. Different reactions. First law of thermodynamics, second law, zeroth law, you have studied. So that thermodynamics we are going to apply in the biological macromolecules. So basic terms you should be knowing here. What is cell? Cell it is the smallest unit which can live on its own and makes up living organism. It is an living organism. So cells are structural, functional and biological unit of living beings. So, what are the basic things we are going to study? This whole unit will be of basic terms. In that, whatever the importance you are going to study in this unit, and what is cell and its constituents, what are the present inside the cell, and next is the molecular basis of life. How day by day the molecules will be interaction in daily routine. So colloids, what are colloids and their classification of colloids and properties of colloidal solution, coagulation of colloidal solution and size of biopolymers, what are biopolymers and methods determining particle shape and their molecular weight. So what are the methods you are going to use to determine the particle shape and molecular weight of that chemical. So also the interaction with water. So hydrophobic and interactions we are going to study here. Before getting to topic, we should be knowing some of the basic terms. Here, what is cell? Cell are structural and functional biological units of living beings. So a cell can replicate itself independently. So it can mutual or it can double itself. So the building it is a building block of life. So this is about the cell and next and the general terms. What are colloids? A colloid is a mixture in which one substance consisting of micro physically dispersed insoluble particle is suspended throughout another substance. So it is one substance substituted in another substance. So for example, colloidal sugar and water, you can take it like that salt and water. So whether it is miscible or not, you can identify. So even the cells have the nucleus and the mitochondria. So what are the constituents present inside the cell andaga? The nucleus, membrane, cytoplasma, mitochondria. These are present inside the cell, lysosomes. So these are the basic terms. And next, cell and its constituents. A cell is a basic unit of living organism. Cells are typically divided into two main categories. So there are two main categories in cells, such as prokaryotic cells and eukaryotic cells. Prokaryotic cells, which are found in bacteria, and eukaryotic cells which are found in all the other organisms. And eukaryotic cells are typically large, more complex, and protein cells are contain specially structures such as nucleus and mitochondria. And cells are composed of various types of biomolecules, including protein, nucleic acid, carbohydrates, lipids. So these are present inside the cells. And coming to the proteins, proteins are nothing but the building blocks of amino acids. We have 21 amino acids, Allah. So these are with the different combination, the proteins will be built. So proteins are the building blocks of amino acids. Proteins are most versatile biomolecule in cells, perform many different functions such as enzymes, structural components, and significant molecules. And nucleic acids such as DNA and RNA are present in the store genetic formation. So nucleic acid such as RNA and DNA strands will be present. And what are the constituents present in that cell andre? Cells, prokaryotic and eukaryotic cells. Prokaryotic cells have no nucleus, and some prokaryotic such as bacteria have region with its cells, and eukaryotic cells are characterized by true nucleus. The size of the cell range between 10 to 100 micrometer in diameter. So, and cell organelles and their functions. What are the constituents present inside the cell? And the nucleus, and nuclear membrane, chromosomes, endoplasmic reticula, globin, and ribosomes. These are the constituents of cells. Mitochondria, lysosomes, chloroplast, vascles are present. So go through the definitions, and these are the basic terms you should be knowing. And next, what are molecular basis of life? The molecular basis of life refers to a fundamental chemical and physical process that occurs within the living organism. So it is an fundamental chemical and physical process that occurs within living organism. So these are known as molecular basis of life. And biological matter is compiled of small molecules. Biological matters is comprised of small molecules, and macromolecules are the it's of large molecules are biopolymers or supramolecules that can be called as macromolecules. And biological matters is comprised of small molecules and intramolecular. And you can be next you will be going to study the interactions and the reactions, how the interaction between the biological system will be taking place. So there will be different types of intramolecular force of attraction. So intramolecular interactions between macromolecules are most conducive to formation of supramolecular structures such as protein complex. Proteins are nothing but the building blocks of amino acids. So they exist in wide variety of physical interactions, and also charge interaction, charge dipole, charge multiple dipole, multiple interactions, and electrodynamic interaction, hydrogen bond interaction, and hydrophobic interaction, intramolecular interaction. There are different types of interaction. In the further units you will be going to know each terms what we are going to use it. So here there are basic definitions for that. Please go through that. And next is a collide. Collides. A collide is a type of mixture in which one substance is dispersed throughout another substance in a small particle. So collides can be classified based on several factors. Collides under one substance will be dispersed in another. So you can take it in water and sugar, salt and water. So it should be dispersed equally. So there should be no aggregation or sedimentation taking place, such as, and it can be colloids can be classified based on several factors, including physical state and dispersed and continuous phase in the type of force holding the practic together. For example, this is an example for stable colloid. This example for unstable colloid. Say there is the particles are not equally distributed inside the substance. So here it is equally distributed. It's not distributed. Aggregation or sedimentation. Aggregation means it is coming upwards and sedimented, which is settling down. So this can be seen here. This is the example for colloids. Colloids can be classified based on the physical state in the dispersed and continuous phase, as well as type of force handling on the particles together. So types of force under aggregation or sedimentation force will be taking place. So some of the examples for colloids under include emulsion, foam, suspension, and translucence because of Tyndall effect. And this can be, and next is the classification of colloids based on the physical state. So the classification is made on physical state. So solid in liquid, where solid particles are dispersed in liquid phase. For example, male, so liquid in liquid, when both the substance are in liquid. For example, oil and water, milk and water, like that. And also gas in liquid, it will be example for carbonated beverages. So you are going to insert carbon dioxide in the water, so that can be a gas in liquid. Solid in gas, for example, smoke, small. Solid in gas, and liquid in gas, liquid which is pressurized, so that can be in the fog. And also gas in solid, a natural gas in the porous rock. So some of the gas liberating from the porous rock, and based on the natural of forcing holding. So these are the types uh, where classification of colloids can be done, and colloids can be classified as follows here. The dispersion ion, that is gas. No such chlorides are known. For example, fog, clouds, gas. For example, here they have taken gas. The medium is dispersion ion medium is gas, gas with gas, and gas with the liquid. And also next is gas with the solid. They are given some of the example: gas with solid, smoke. They are given gas with liquid. They are given fog, cloud. So some of the substance how it with the different classification of colloids are seen. So liquid with gas, this one. Liquid with, see, this is the different terms. Here they taken gas, liquid in the second column, and third column, that is with respect to solid. So liquid with gas, that is foam. Liquid, that is emulsion, milk, mayonnaise. So the those are for liquid with liquid. Liquid with solid, ink, pigmented ink, sedimented. So solid with gas, aerosol, floating soap. And solid with, some of the examples they are given: solid with liquid, that is gel, and solid with solid, carbonated gas, glass. And next, interaction between particles. So some of the force plays an important role in the interaction of the colloidal particles. So there will be force acting on the colloidal particle with different substances. So exclude volume repulsion. If the volume is excluded, there will be repulsion in the volume. So this refers to impossibility of overlap between the art particles. and electrostatic interactions. So here colloidal particles often carry an electrical charge, therefore attract, repel each other. So some of the colloidal particles will be having electrical charges, so there will be attraction or repulsion between the colloids. This can be and different types of interaction. Van der Waals force of interaction, steric interaction, hindrance, it can be seen in the colloidal particles also. This is due to interaction between two dipole that are either permanent or induced. Van der Waals force of interaction and steric force of interaction. So next, properties of colloidal solution. Colloidal solution is also known as a colloid. It is a type of mixture in which one substance is dispersed throughout another substance. So colloids andre it is nothing but one substance dispersed in another substance, whether it may be liquid, liquid, solid, liquid, gas, liquid. So it may be in the different phases. So depending on the different physical property, the colloids can be classified here. And here coming to the properties, it is polarization can be classified based on the types of force holding the particles together. So it can be electrostatic force of interaction, Van der Waals force of interaction, and also with respect to chemical bond formation. Uh, they can also be classified based on the physical state. Mainly, it is classified on the physical state and continuous phase, such as solid in liquid, liquid in liquid, gas in liquid. Colloidal solution have unique properties that differs from those of true solution and suspensions. So different type effects you can be seen a Tyndall effect when a beam of so you go through these terms, ನಿಮ್ಗೆ each term ಇದು ಓದಿದ್ರೆ you will be getting to know the filtration ಅಂದ್ರೆ ಏನು, sedimentation ಅಂದ್ರೆ ಏನು, light scattering, electrophotic mobility, stability, high viscosity, high surface area of volume, Brownian motion, Tyndall effect. So Tyndall effect ಅಂದ್ರೆ when the light is scattered by the dispersed particle. So when the substance one type of substance is dispersed in another substance, when light is passed through, the light gets scattered inside the media, so that can be known as Tyndall effect. And these are the terms you should be knowing. And also with respect to high viscosity, ಅಂದ್ರೆ the colloidal solution tends to have higher viscosity than true solution or suspension due to increase in number of particles. When the number of particles are increased, there will be the viscosity will be increased. And also filtration colloidal can be difficult to filter as the particles are too small to be trapped by the filtration. So these are the basic terms. And next, coagulation of colloidal solution. Coagulation is the process by which colloid solution become less stable and disperse particle start to aggregate and form large particles. So it is the process by which the colloidal solution become less stable and disperse the particle and aggregate to form the large particle. There are different factors which are affecting the coagulation. So for example, coagulation of blood, you can be seen. Also here they are given some of the factors which affecting the coagulation. So addition of an electrolyte. Coagulation ಯಾವಾಗ ಆಗುತ್ತೆ ಅಂದ್ರೆ addition of an electrolyte. Electro can neutralize the charge on the dispersed particle. And change in pH. A change in pH can alter changes of dispersed particle. So why these are the changes going to take place andre due to when you are going to add the substance from different substance ಈಗ react ಆದಾಗ, so the interactions everything will be seen. So when you form a formation of colloids also, they can see some of the interactions also change in their physical state. So addition of electrolyte, change in pH, increase in temperature. When some of the interaction is taking place, the reaction is taking place, andre, the increase in temperature can cause solvent to evaporate. Means the interaction is taking place, andre, definitely the increase in temperature take place. Addition of coagulate, certain substance can be added while the reaction is taking place, and shear caused by mechanical agitation. So these are the terms. And sedimentation velocity andre, when it is getting settled down. So it is a technique used in the biochemistry and biophysics to determine the size or shape and weight of macromolecules such as proteins and nucleic acid. Macromolecules andre proteins and nucleic acid. So what are the examples for macromolecules andre? Then it's nothing but the size of the molecules are big. So here it is proteins and nucleic acid having DNA and RNA strands. Proteins andre, it is a different combinations of amino acids. Amino acids, there are 21 amino acids. So on different combinations, the proteins will be formed. So the technique is based on the principle that larger and heavier molecules will sediment. Means settling down. The larger and heavier molecules, it will settle down when it is dispersed in a media. It will settle down. So larger and heavier molecules. By measuring the uh, sedimentation velocity, how you are going to, why you are going to measure andre, to determine the size, shape and weight of that macromolecules. So that we are going to measure by measuring the rate at which the macromolecule sediment. The rate at which the macromolecule will sediment, if you calculate, we can measure, determine the size, shape and weight of the macromolecules. So the rate at which the particles of a given size, the rate at which the particles of a given size, shape will sediment in centrifugal field. So centrifugal field are in the big. So when it is centrifuged, the sedimented or heavy molecule will settle down. Centrifuge ಮಾಡಿದಾಗ. So the formula we are going to calculate the sedimentation velocity ಅಂದ್ರೆ V is equal to 2r square G into D by 10 nr. So V is the sedimentation velocity. N is the viscosity of the solvent. G is the acceleration due to gravity. So that is with respect to centrifuge. And D is the density of that particle. So next is the biopolymers. Biopolymers are type of polymers that are naturally occurring or synthesized by living organism. Polymers ko biopolymers ko there is a difference. Polymers andre a class of natural or synthetic substance. Natural or synthetic substance composed very large molecules are called polymers. So biopolymers andre it is of living organisms. Biopolymers are the type of polymers that are naturally occurring or synthesized by living organisms. So those are called as biopolymers. They can be comprised of various monomers such as amino acids, sugars, nucleotides. They are made up of. And although biopolymers are proteins, nucleic acid, such as DNA and RNA, and also different types of polysaccharides. So these are also known as biopolymers. Proteins are biopolymers composed of amino acids perform a wide range of functions in living organism. So it performs wide range of functions in the living organism. Proteins. What are the proteins? Proteins are nothing but building blocks of amino acids. So it will be of macromolecules and the size will be of larger. So this can be also known for biopolymers. Biopolymers ಅಂದ್ರೆ it is synthesized by living organisms. So nucleic acids are such as DNA and RNA are biopolymer composed of nucleotides or carry genetic informations. So polysaccharides are biopolymer composed of sugar are important energy storage of molecules in many organisms. They include glycogen, cellulose, and creatine. And this is the structure of protein. So, or also it can be known as biopolymer. Protein andre it is having uh, of amino acid. It's nothing but the building blocks of amino acids. So then in the next unit you'll be knowing what are amino acids and its constants. So how the proteins will be built. So this is an example structure for protein. Protein can range in size from small peptide containing just few amino acids to large complex containing hundreds of amino acids. So it will be up to thousands of amino acids in the protein structure. Uh, Smallest known protein can called be peptide. So where largest known protein which will be having up to 34,000 amino acids. ಈ protein structure ಅಲ್ಲಿ up to largest ಅಂದ್ರೆ 34 34,000 amino acids will be present. Smallest ಅಂದ್ರೆ peptide it will be having 4 to 5 amino acids. Peptide also known as the building block of amino acids which is having very less, that is of 4 to 5 amino acids. And the largest protein structure andre it will be having 34,000 amino acids. So nucleic acid, next nucleic acids such as DNA and RNA will be present. So it will be having around 3 billion base pair of DNA, and the pairs andre 3 billion base pairs will be present. DNA can be present. And also the size of polymers, which can be determined by various techniques such as gel electrophoresis, chromatographic technique, and spectrophotometry techniques. The size of the biopolymers can be measured using gel electrophoresis, chromatography, and mass spectrophotometry technique. So, methods for determining the particle shape and molecular weight. So it can provide important information. Why you need to determine the particle size and molecular weight of that substance or molecules ಅಂದಾಗ it can provide important information about physical and chemical properties of that substance. Why you are going to determine molecular weight and physical shape of that ಅಂದ್ರೆ to physical and chemical properties na identify ಮಾಡಕ್ಕೆ. So molecular weight is a measure of mass of molecules. So it is molecular weight is nothing but the measure of mass of molecules, and it can be used to calculate the density and other properties of substance. So there are several methods for determining the shape and molecular weight. Uh, so such methods, there are some few instrumentations how to determine the shape and their molecular weight. So shape can be determined by SEM or TEM. So such as scanning electron microscopy or TEM transmission electron microscopy can be used to determine the shape of the substance. So light scattering method it can be used to determine the size of the molecules. ಇಲ್ಲಿ size of the molecule determine ಮಾಡಕ್ಕೆ we are going to use LS method, that is light scattering method or dynamic light scattering method. So these are also instrumentation where we are going to use different instruments to calculate the size of the given molecule which will be unknown. So and shape of that molecule to identify ಮಾಡಕ್ಕೆ we are going to use the instruments such as scanning electron microscope and transmission electron microscope determine the shape of determine ಮಾಡಕ್ಕೆ. And also we can use the X-ray crystallography machine to crystalline structure or molecular weight. More than it can mainly used for determine the crystal structure. X-ray diffraction, that is nothing but X-ray crystallography. So and also mass spectrometry, the method is used to charge ratio of ions to determine the molecular weight. Molecular weight use ಮಾಡೋದಕ್ಕೆ mass spectrophotometry we are going to use. And also another method we are going to use NMR technique to determine the mass uh, molecule and to determine the structure of molecule and their molecular weight. It can be determined. So there are several methods to determine the molecular weight and biochemical, such as gel permission, fermentation, chromatography. This method is used to column field. So chromatography technique you have studied. It is mainly used to separate the substance to identify the molecular weight. So size exclusion chromatography. This method is similar to GPC but used to different types of column. It separates the molecule based on their sites. So mainly these chromatography techniques are used to separate the molecules. And NMR technique is used to find the structure of molecule and their molecular weight. So each instrument, what is the purpose you are going to use, and you should be knowing each instrument. So NMR, UV visible spectroscopy, mass spectroscopy, and so these are the instrumentation methods which are going to identify their shape, molecular weight, size, and next is the interaction with water. So here biopolymers. What are biopolymers? Biopolymers ಅಂದ್ರೆ is nothing but the biopolymers ಅಂದ್ರೆ ಈಗ ಹೇಳಿದ್ದ the type of the polymers that are naturally occurring and are synthesized by living organisms. For example, proteins, nucleic acid, polysaccharides. These are biopolymers. Biopolymers are type of polymers that are naturally occurring and are synthesized of living organisms. Again, why I am repeating andre you should be knowing these terms. Further biopolymer ಅಂತ next ಬರುತ್ತೆ ಅಷ್ಟೇ. Like ಮತ್ತೆ definition ಹೇಳ್ತಿರಲ್ಲ. So protein, the interactions, how it interacts with the water. So biopolymers such as proteins, polysaccharides, nucleic acids, how it's mainly because why it is they are concentrating with respect to water andre mainly because the proteins are present inside the body. So the body is having 70% of water. So there will be an interaction between the protein water molecule. So that's why we are going to study the interaction in the with the water molecule. So proteins generally where you are going to find protein, nucleic acid, polysaccharides are present inside the body. So the interaction between the proteins and water you are going to study in this. Why and in the interaction with water, the biopolymers such as protein, polysaccharides, nucleic acid has specific interaction with water. They are important for their function and their stability. Proteins are comprised of amino acid which have both hydrophobic and hydrophilic nature. Hydrophobic ಅಂದ್ರೆ water repelling, hydrophilic ಅಂದ್ರೆ water attracting. So this should be knowing hydrophobic interaction and hydrophilic interaction ಅಂದ್ರೆ ಏನು ಅಂತ. Water repelling and water attracting. The hydrophobic region of protein will tend to interact with each other, avoid water. The hydrophilic region will interact with water. So hydrophobic, hydrophilic difference ಗೊತ್ತಿದ್ರೆ ಸಾಕು. If you are knowing the terms, hydrophobic ಅಂದ್ರೆ water repelling, hydrophilic ಅಂದ್ರೆ water attracting. So based on that, you are going to name whether it is having hydrophilic nature or hydrophobic nature. And also biopolymers interact with water, plays a major role in the solubility, stability, and also functionality. Understanding these interactions which may help in the design and optimize the biopolymer based materials. So this is with respect to first unit, cell and constituents of biopolymers, so proteins, so nucleic acid. These are with respect to first unit. And in the second unit, we are going to study about molecular interpretations of energy and enthalpy. So you should be knowing energy and enthalpy ನೀವು ಓದಿದ್ದೀರಾ in the thermodynamics. There we are studied thermodynamics, kinetic energy, potential energy, enthalpy, and entropy. ಓದಿದ್ರ ಅಲ್ವಾ? ಎಂಥಾಲ್ಪಿ ಅಂದ್ರೆ ಇಟ್ ಈಸ್ ದ ಸಮ್ ಆಫ್ ಇಂಟರ್ನಲ್ ಎನರ್ಜಿ and pressure, time, volume. Entropy is measurement of system's thermal energy. Entropy is nothing but with respect to temperature. Entropy andre. So thermodynamics ಓದಿದ್ದೀರಾ? So thermodynamics, it is the flow of heat. So we are going to study the physical and chemical process taking place inside that system. Thermodynamics ಅಲ್ಲಿ. And also kinetic energy, it is a form of energy that an object or a particle which has reason for its motion. So potential energy andre it is nothing but stored energy. Stored energy, it can be named the potential energy. So basic terms ಈ unit ಅಲ್ಲಿ again you should be memorizing ಅಂದ್ರೆ enthalpy and entropy. Enthalpy andre it is the sum of system internal energy. Entropy andre it is with respect to temperature, the measure of system's thermal energy. So main topics we are going to study in this unit andre the molecular interpretation of energy and enthalpy. Enthalpy andre the sum of internal energy, and non-covalent reactions, covalent and non-covalent reactions, and hydrophobic interaction, biochemical applications of thermodynamics, thermodynamics for metabolism, double strand formation of nucleic acid, ionic effect on protein and nucleic acid. So previously we have taken in thermodynamics what we have done andre we used to take a general reaction and study inside the system when the reaction is taking place, whether uh, the energy is liberated or absorbed by the system, the temperature is generated or absorbed by the system. So what are the physical uh, process it is taking place, physical or chemical process it is taking place, we are going to study ಅಲ್ಲ, keeping volume constant, keeping pressure constant, whether the reaction is taking place or not, we are going to study in the thermodynamics. Same thing we are going to apply in this biopolymers. So with the living organisms we are going to study whether the reaction is taking place, whether it is having any interaction with that Means illo ಅಷ್ಟೇ ಅಲ್ಲ ಸೇಮ್ ಥಿಂಗ್ ದ ಪ್ರೋಟೀನ್ಸ್ ಅಂದ್ರೆ it is nothing but living systems where it's a building block of amino acids. So amino acids ಈಗ 21 amino acids ನೀವು ಸ್ಟ್ರಕ್ಚರ್ಸ್ ನೋಡ್ತಾ ಹೋದ್ರೆ it is nothing but a chemical structures. ಅದೇನ್ ಇನ್ನೇನಿಲ್ಲ. Lysine, glycine, 21 amino acids are there. So that one I will send you the slide with all the informations. Mm, So amino acids are proteins are building blocks of amino acids. Amino acids continuous chain structures say this formation of proteins. So the how the interaction here the proteins with the different substances with the water molecule we are going to study. So here also we are going to apply thermodynamics uh, to study the heat and other forms of energies. So it depends mainly for chemical reaction occurs and how the energy is transferred during the reactions. It can be studied in this unit. So molecular interpretations of energy and enthalpy. Those also can be studied. So here coming to the topic in thermodynamics is a branch of chemistry that deals with relationship between heat and other forms of energy. So it is how the chemical reaction occurs, how energy is transferred during the reaction. So it can be studied in this. And protein nucleic acid interaction play key role in many biological process. Protein nucleic acid interaction play a key role in biological process. This is very much important main thing. Protein nucleic acid interaction plays key role in many biological process. So protein, you are already knowing that. So what is protein, Andre? It is an building blocks of amino acid. So the group of amino acid forms the protein structure. And nucleic acid, Andre, it is a naturally occurring chemical compounds that serves as a primary information and carrying of molecules in the cells. It is nothing but chemical compound, nucleic acid and carrying molecules in cells. So that is what we are going to study. Protein and nucleic acid interaction plays important role in the biological process. So biological interact with protein interacts with DNA and RNA through similar physical force, which includes there will be formation of interaction. If the two substance react together, there will be formation of energy salva, such as there will be formation interaction will be formed, including electrostatic interaction, salt bridge, there will be formation, dipole interaction, nitrogen bond interaction, entropy interaction, hydrophobic interaction, hydrophilic interaction. So these are the interactions which will be formed. When andaga, this interaction will be formed when protein nucleic acid interaction. The protein when react with the nucleic acids. When protein gets in contact with the nucleic acid, they play a key role in many biological process. And molecular interpretation of energy and enthalpy. Energy is the ability to do work. So what is energy ಅಂದ್ರೆ molecular interpretations of energy and enthalpy. Energy ಅಂದ್ರೇನು enthalpy ಅಂದ್ರೇನು ಅಷ್ಟೇ. So energy is the ability to do work or cause for the change in the energy. In the molecule level, energy can take many forms such as kinetic energy, potential energy, chemical energy. thermal energy. These are with respect to energy. Enthalpy andre enthalpy is a measure of total energy of the system. Enthalpy and entropy you should be knowing. Enthalpy andre it is a measure of total energy of the system. Initial state to final state anta barutte. Initial state andre the when the reaction is started adu initial state, when the reaction is completed that is the final state. So both the thing we are going to take initial state and final state. Enthalpy E naught and E1 andre that will be enthalpy at the initial state, enthalpy at the final state. Now we are going to calculate enthalpy anta bandaga enthalpy final state minus initial state. The reaction ಆಗಕ್ಕೂ ಮುಂಚೆ reaction ಆದ್ಮೇಲೆ what is the energy, whether the energy is liberated or absorbed by the reaction, it can be seen. So for example, energy delta G that will be of Gibbs free energy. The previous you have studied Gibbs free energy ಏನು ಹೇಳುತ್ತೆ when delta G is equal to 0 ಅಂದ್ರೆ enthalpy free energy is 0 ಅಂದ್ರೆ the reaction is in equilibrium ಅಂತ, where there is no reaction taking place or reaction taking place andre it will be in the equilibrium state. When the delta G is in negative andre the reaction is in spontaneous process. ಡೆಲ್ಟಾ ಜಿ ನೆಗೆಟಿವ್ ಅಂದ್ರೆ ಫ್ರೀ ಎನರ್ಜಿ ನೆಗೆಟಿವ್ ರಿಡ್ಯೂಸ್ ಆಗ್ತಾ ಬರ್ತಿದೆ ಅಂದ್ರೆ ಗಿಬ್ಸ್ ಫ್ರೀ ಎನರ್ಜಿ ರಿಡ್ಯೂಸ್ ಆಗ್ತಾ ಬರ್ತಿದೆ ಅಂದ್ರೆ ರಿಯಾಕ್ಷನ್ ಈಸ್ ಟೇಕಿಂಗ್ ಪ್ಲೇಸ್. ಇಟ್ ಈಸ್ ಕನ್ಸ್ಯೂಮಿಂಗ್ ದ ಎನರ್ಜಿ ಟು ಫಾರ್ಮ್ ದ ಪ್ರಾಡಕ್ಟ್ ಫಾರ್ಮ್ ಆಗೋದಕ್ಕೆ ಎನರ್ಜಿ ಕನ್ಸ್ಯೂಮ್ ಆಗ್ತಿದೆ. ಸೊ ಎನರ್ಜಿ ವಿಲ್ ಬಿ ಗೆಟಿಂಗ್ ರಿಡ್ಯೂಸ್ ಇನ್ಸೈಡ್ ದ ಸಿಸ್ಟಮ್ ಒಳಗಿರೋ ಎನರ್ಜಿ ಕಡಿಮೆ ಆಗ್ತಾ ಬಂದ್ರೆ ದ ರಿಯಾಕ್ಷನ್ ಈಸ್ ಟೇಕಿಂಗ್ ಪ್ಲೇಸ್ ಅಂಡ್ ದ ರಿಯಾಕ್ಷನ್ ಕ್ಯಾನ್ ಬಿ ನೋನ್ ಫಾರ್ ಸ್ಪಾಂಟೇನಿಯಸ್ ಪ್ರೋಸೆಸ್. ನಾನ್ ಸ್ಪಾಂಟೇನಿಯಸ್ ಪ್ರೋಸೆಸ್ ಯಾವಾಗ ಆಗುತ್ತೆ ಅಂದ್ರೆ ವೆನ್ ಡೆಲ್ಟಾ ಜಿ ಈಸ್ ಇನ್ ಪಾಸಿಟಿವ್ ಅಂದ್ರೆ ಎನರ್ಜಿ ಇನ್ಕ್ರೀಸ್ ಆಗ್ತಿದೆ, ಸಿಸ್ಟಮ್ ಅಲ್ಲಿ ಇನ್ಕ್ರೀಸ್ ಆಗ್ತಿದೆ, ರಿಯಾಕ್ಷನ್ ಫಾರ್ಮ್ ಆಗ್ಬೇಕಾದ್ರೆ ಸಿಸ್ಟಮ್ ಎನರ್ಜಿ ಇನ್ಕ್ರೀಸ್ ಆಗ್ತಿದೆ ಅಂದ್ರೆ ದ ರಿಯಾಕ್ಷನ್ ಈಸ್ ನಾಟ್ ಟೇಕಿಂಗ್ ಪ್ಲೇಸ್ ಅಂತ. ದೇರ್ ಇಸ್ ನೋ ಚೇಂಜಸ್ ಇನ್ ದ ಡೆಲ್ಟಾ ಜಿ ಆರ್ ಅಂದಾಗ ದ ರಿಯಾಕ್ಷನ್ ಈಸ್ ನಾಟ್ ಟೇಕಿಂಗ್ ಪ್ಲೇಸ್. ಸೊ ಡೆಲ್ಟಾ ಜಿ ವಿಲ್ ಬಿ ಇನ್ ದ ಪಾಸಿಟಿವ್. So coming back to this enthalpy, so the molecular interpretation of energy is given by delta E is equal to Q plus W. Delta E is nothing but change in internal energy. ನಾನು ಹೇಳಿದ್ನಲ್ಲ, if there is a change in internal energy, the reaction is taking place. If there is no changes, the delta E will be 0. The reaction equilibrium state ಅಲ್ಲಿ ಇದ್ರೆ. If the positive delta energy delta E is in positive andre the reaction is not taking place or formation of product is not formed anta. So here delta E represent change in internal energy. Q is heat added to the system. W is the work done by the system. So that is the molecular interpretation of energy by given equation. Delta E is equal to Q plus D. So how we are going to calculate the enthalpy andre H enthalpy can be denoted by H is equal to E plus PV. P is the pressure, V is the volume, is E andre internal energy, H andre enthalpy, E andre internal energy. So what is the difference between H is equal to E plus PV and delta H is equal to delta E plus delta illide delta illa? What is the difference? So why? Same formula can be denoted by two forms. the change in enthalpy, the change in energy, the change in pressure or change in volume, it can be denoted by delta. If there is any changes taking place, it can be denoted by delta. So delta H, there will be changes taking place when the reaction is taking place. So that can be denoted by delta. So coming to the enthalpy versus internal energy. So this is, for example, this one we can consider as an system. So inside this, the reaction is taking place. We have placed thermometer to determine the temperature. So this is an old method stirrer anta illide to equally distribute anta. So here in recent years we are going to use magnetic stirrer. Magnetic bit will be inside the reaction. There will be equally stirred. So also connection to this and wire, and this is what the total system, and you can measure the pressure, increase or decrease the pressure for this chamber can also be inserted. So if the no work is done by the system on its surrounding, delta es, delta e system is equal to q, at constant volume. If the volume is supplied constant, whatever the reaction taking place, the equal, ಇದರಲ್ಲಿ ಇನ್ನೂ ಒಂದಿದೆ ಗೊತ್ತಾ, ವಾಟ್ ಇಟ್ ಹ್ಯಾಪನ್ಸ್ ಅಂದ್ರೆ, ಇಫ್ ದ ರಿಯಾಕ್ಟೆಂಟ್ಸ್ ವಾಟ್ ವಿ ಆರ್ ಟೇಕನ್ ಫಾರ್ ದ ರಿಯಾಕ್ಷನ್, ದ ರಿಯಾಕ್ಟೆಂಟ್ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಇನ್ಕ್ರೀಸ್ ಮಾಡಿದ್ರು ಇಟ್ ವಿಲ್ ವೇರಿ, whatever the formation of the product, even the temperature, everything will be varied. So here we are just increasing the volume of the reaction, andre concentration alla, volume we are going to increase. The heat given off or absorbed by the reaction can be seen in the system. So that can be denoted as delta E. Delta E is the system internal energy. E represent the internal energy. That is what ESYS andre systems internal energy. So at constant volume, the heat gives off are absorbed by the system. So delta E system is equal to QV. So for example, calorimetry in which the reaction can be run at constant volume. The volume, the reactants what we are taken, it is in the constant, and the most of the reaction are run in open flask or beaker. It can be run almost closed system alle run ಮಾಡ್ತೀವಿ. There are some of the reaction, for example, closing the beaker or in the jar, it can be done the system. So keeping the thermometer and different holes for the pressure. The system is at constant pressure. However, the total pressure inside the container is always equal to atmospheric pressure. If there is no change in taking place with respect to system and external, that is atmospheric pressure. And the change in enthalpy of the system, same thing they are given here. Change in enthalpy, whatever the can be represented by delta. So the change in enthalpy H is equal to E plus PV, that is for enthalpy. So change in enthalpy that can be determined by delta. So next important is andre, the heat gives off or absorbed when the reaction is run at constant volume is equal to change in internal energy of the system. When the reaction is at constant volume, means the constant, for a system the constant volume is needed ಅಂದ್ರಾಗ it change in the internal energy of the system. So change in internal energy of the system ಅಂದ್ರೆ volume increase ಆದಾಗ there will be change in the internal energy of the system. So change in enthalpy of the system, it can be observed by the system. So that is with respect to delta H system. That is the change in enthalpy of the system. So it can liberate or absorbed by the system. And also next is with respect to change in internal energy of the system. That is delta H SYS, delta E SYS plus system. SYS andre it's nothing but the system. So plus delta PV. So next is the non-covalent reactions. So just brief ಮಾಡಿದ್ರೆ ಸಾಕು ಅನ್ಕೊಂತೀನಿ. There are not much members. I hope so. Non-covalent interactions are weak interactions between atoms and molecules where no chemical interaction taking place. So non-covalent interactions are weak interactions or no reaction taking place between the molecules or atoms. And electrostatic interactions andre they are type of non-covalent interactions, electrostatic interactions also. So next is the hydrophobic interactions. Hydrophobic interaction describe the relation between water and hydrophobes. So hydrophobes are nothing but physical properties of molecules. Molecules na you are going to represent in the hydrophobes. Hydrophobes andre they are such nothing but the molecules, and hydrophobes are non-polar molecule usually have long chains of carbon that do not interact with water. And for example, they are given here hydrophobic hydrophobes andre they will interact with total here 8 molecules and plus here 8. What will be the total? ಎಷ್ಟಾಗುತ್ತೆ? There will be 16 molecules. The formation will be only of 10 atoms after the interaction. So the interact with total, the indicates the hydrophobes come together. They will have less contact with the water. And also thermodynamics for hydrophobic interactions. Thermodynamics for hydrophobic interaction andre when hydrophobes is dropped in an aqueous medium, the hydrogen bond between water molecules will break and make room for hydrophobe. However, water molecules do not react with hydrophobes. So here hydrophobic interaction we are going to calculate with respect to thermodynamics. They are said in the next. The hydrogen bond broken by entrance of hydrophobe that can be seen where with respect to water molecule. This an hydrophobe, and this is with respect to blue color that is represented as hydrogen bond. So when hydrophobes interact, the hydrogen bonds broken by the entrance of the hydrophobes. There will be change in enthalpy. So that can be represented. How you are going to represent andre delta G is equal to delta H minus T delta S. So delta G is nothing but the Gibbs free energy. Delta H andre enthalpy of the system. T is the temperature. S is the pressure. So the Gibbs free energy is in positive andre that we have discussed. I have told you again. When the Gibbs free energy is in zero means that is in the equilibrium state. When it is in the negative andre, as the reaction is taking place, when delta G is in positive, the reaction is not taking place. So, and next is the strength of hydrophobic interactions. Hydrophobic interaction, the strength of hydrophobic depends on the several factors. Hydrophobic or hydrophilic interaction, you should be knowing what is the difference between hydrophobic and hydrophilic interactions. So in hydrophobic interaction, the temperature increases, the strength of hydrophobic increases. Uh, these are the factors which are interpreting the hydrophobic interactions on the and number of carbons on the hydrophobes. Molecules with greater number of carbons will have the strongest hydrophobic interactions, and also the shape of hydrophobes. Aliphatic organic molecules are strong interactions, uh, and aromatic compared to the aromatic. Aliphatic andre it is nothing but open chain structure. Aromatic andre it is of closed chain structure. So that is with respect to aliphatic and aromatic organic molecules. So next is biochemical applications of thermodynamics. So what are the applications? Thermodynamics andre in biochemistry, the thermodynamics used to study. See what is the concept here? It we going to know by the thermodynamics used to study the understand the biological process such as enzyme activity, protein folding, and chemical reactions. Thermodynamics we are going to implement in this biochemistry or biological ಇದಕ್ಕೆ ಅಂದಾಗ we need to will be getting to study about enzyme activity, protein folding, and also with respect to chemical reactions. It can be studied. The principle of thermodynamics can be used to predict the direction and rate of reaction and also the stability of the molecule and their complexes that can be studied using the thermodynamics. And coming to the first law of thermodynamics, that is, energy can neither be created nor be destroyed. Uh, but here, the energy is in the closed system. Energy liberated or observed that will be in the closed system, the reaction will be taking. So, energy will remains constant. And while applying second law of thermodynamics, energy is transferred and it will be less energy available at the end of the transfer process. So entropy increases as energy is transferred. Entropy andre no. Entropy is nothing but the measure of system's thermal energy. So as the entropy is transferred, as the as the energy is transferred, the entropy gets increased. Energy transfer adre, the entropy will get increased, and the entropy is nothing but the system thermal energy. The temperature also will get increased. And next, the biological importance of hydrophobic interactions. What are the biological importance? So they are important in the folding of proteins. Protein foldings andre nothing but see here. You can see the foldings. ಈಗ one strand ಬಂದ ತಕ್ಷಣ it is folding in the next. So that will be actions. This is important to keep protein stable and biologically active because it allows to protein or decrease the surface area and reduce the interaction with water molecules that can be seen. And the relation between thermodynamics and metabolism. So thermodynamics used to study the transfer of energy in living organism and how it is used to drive a chemical reaction in the body. So how the chemical reaction will be taking place inside the body that can be studied, and also the transfer of energy inside the living organism that can be studied using thermodynamics. So delta G is equal to delta H minus T delta Delta G is the Gibbs free energy, delta is the enthalpy, and T is the temperature, S is the entropy of the system. So that these are the basic terms, and this is the double strand formation of nucleic acid, that is of DNA and RNA, that is known as nucleotide. So the double strand formation is important for the stability and function of nucleic acid. So here the DNA double helix biopolymer nucleic acid is held together by nucleotide which base pair together. So many proteins which bind to DNA. So through the wide major group, many proteins with bind with the DNA or double helix structures of DNA molecule. So what is ion effective on protein nucleic acid interaction? Protein nucleic acid interaction ಅಂದ್ರೆ proteins with the nucleic acid, there will be formation of interaction. So ionic chains can have significant effect on protein nucleic interaction. If the ionic strength is high, the electrostatic interaction between the protein and nucleic acids are weak or very less. If the ionic strength ಏನಾದ್ರು high ಇದ್ರೆ the electrostatic interactions between protein and nucleic acids are very less. If the ionic strength is in less ಅಂದ್ರೆ the electrostatic interaction are strong. So this is the difference between the ionic effect. Means if the ionic strength is high, the interaction will be less. If the ionic strength is less, the interaction will be more. That can be denoted by delta H is equal to delta G is equal to delta H minus T delta S. Delta H is for enthalpy change. Here we can calculate electrostatic interaction, hydrogen bonds, van der Waals contract, and distribution of torsion angles. Here coming to delta S, that is with respect to entropy change. The hydrophobic interaction and rotation and transition, conformation, dynamic, and counter ion release can be studied. So these kings, everything will can be studied for these structures with keeping the formula delta G is equal to delta H minus T delta S. So what are the key factors influencing enthalpy, entropy, and free energy? So formation of protein nucleic acid formation ಆಗ್ಬೇಕಾದ್ರೆ, so what are the key factors affecting ಅಂದ್ರೆ the hydrophobic effect that accompany a decrease in nonpolar surface area exposed to the solvent. So hydrophobic effect can be seen. Also the rotation and transition restriction of interacting macromolecules. There will be restriction in the rotation of the macromolecules. Macromolecule ಅಂದ್ರೆ very big size in the molecular structure ಅಲ್ಲ. So the there be some restrictions. This can be also known for macromolecules. So also conformational dynamics and counter ion release in the formation of the reaction. So these are the key factors that influences the enthalpy, entropy, and free energy. Enthalpy is nothing but the sum of internal energy. Entropy is the measure of system's thermal energy. They should be knowing. This is the end of the unit.